And I would like to welcome our second speaker of this session, and that's Charlie Scaff. Charlie did a PhD in Warwick and explored various things with structural mass spectrometry. And then afterwards, she moved on to the University of Leeds, to the Asbury Center, where she did her postdoctoral research. And then in 2017, she was awarded an ISSF fellowship. And now in 2020, she became a BHF fellow. And in 2022, she was awarded a university academic fellow in structural biology and is doing work now, not just with structural mass spectrometry, but also combines with biochemistry and also cryoEM, as well as HDX. And we're really excited to hear all about these things. Thank you.
Thank you very much for the introduction, and thank you very much for the invitation to speak here today about our work using time-resolved cryoEM to capture myosin in action. And this is a collaboration between my group, who largely work on the structural biology of myosin, the group of Steve Munch, with his interest in the development of sample preparation and time-resolved cryoEM-based techniques, and Howard White, who is a myosin kineticist. And this work was largely completed by two very talented PhD students, David Clareball, who's now moved on to work with John Briggs at Martins Reed, and Sean McMillan, who is within my team, but is currently writing up his PhD thesis. So hi, Sean, and hopefully I can do this work justice. Okay, so I work across the Leeds Institute for Cardiovascular and Metabolic Medicine, where we have an interest in how myosin and muscle is involved in disease, and the Asbury Centre, supported by a wonderful Asbury Biostructure Laboratory with two Titan Crioses, FibSem, and a wonderful support team, where we work to understand life in molecular detail. And this is where time-resolved comes in, because if we want to understand how biological systems work on the molecular level, structure does not equal function alone. We need to understand how these things move to undergo their dynamic processes. And so if we look at these various snapshots of a horse galloping here, just capturing those snapshots is not enough to understand how it functions. We need to be able to put them in order. And we can do that by using time. And that allows us to recapitulate this biological system in action. And this is never any more important than in the world of molecular motors, like this ATPase here, or in the world of myosin, which is my favorite topic. So myosins are actin-based molecular motors, and they're responsible for force generation and movement within our cells, fueled by ATP hydrolysis. They're essential to life, and as such, involved in many diseases, from cancer to heart disease. And although they have multiple different functions, they have a very conserved domain structure with a head and a tail region. And the head comprises of the motor domain, which binds ATP and uses the energy produced from ATP hydrolysis to move a lever and transmit force. So whether we're talking about myosin 2, that assembles into thick filaments in our muscle to drive contraction, or myosin 5, a two-headed motor that transports vesicles by walking along actin filaments, they still have the same conserved chemomechanical cycle, fueled by the energy of ATP to drive contraction or motion. And you might remember this from your biochemical textbooks. So we've got a motor domain and a lever, and the lever swings to generate force. And this was what you'll find in your textbooks. In Rigor state, active with no nucleotide present, actin is bound to myosin. We add ATP that dissociates the myosin the ATP is hydrolyzed to produce a lever up state that's undergone the recovery stroke that can then bind to actin and release phosphate, generate the power stroke, and result in a post-power stroke lever down. So we go from a primed lever up position, we undergo the power stroke, termed the swinging lever hypothesis, and we end with a post-power stroke lever down state. And this is what you'll find in your textbooks. However, before we did this work, there was no pre-power stroke primed actin-myosin state in existence. So no one had actually been able to demonstrate the swinging lever mechanism. We had lots of off-actin states captured by crystallography, and we had some strongly bound post-power stroke states with the lever down, captured by cryoEM, but this remained elusive, and so we couldn't demonstrate this mechanism. And this was largely because the turnover of this state is on the millisecond time scale. So how do you trap it? Well, we've managed to do that using time-resolved cryoEM. And the late Professor John Trinick and Howard White actually came up with this experiment around 40 years ago. And they developed this initial plunge freezing device that was super fast with a pneumatic plunge freezer to try to trap this state. But unfortunately, they didn't have the microscope advances that we now have to bring this state to fruition. So it's taken till now with advances in time-resolved freezing apparatus, as well as microscope detectors and image processing, etc. So Steve Munch's sample prep team and the help of a lot of individuals have taken this initial time-resolved device forward to produce this time-resolved spraying device now, where we have a sprayer which sprays out your sample. You have your grid suspended in tweezers above, and then you rapidly plunge your grid through the spray and into the liquid ethane. And this is the latest device here with a microfluidic sprayer. So we can mix two things very quickly. We produce a fine droplet spray. We have our grid in the tweezers. We use self-wicking grids. And you can see in the movie here, this is going to move through the spray very quickly and then go straight into the liquid ethane. Obviously, this movie is slowed down, and we can go sort of around 10 milliseconds timescale. And this has now allowed Steve's team to produce lots of structures to high resolution. But what we're interested in doing is applying it to looking at this actin-myosin state. And we chose an actin-myosin 5 because Howard has worked on this for about 40 years. We understand the kinetics of this cycle very well. And we used a mutant that basically increased the number of molecules in this primed actin-myosin state, so we had a better chance of capturing it. And the experiment design looks a little bit like this. So we pre-mix the myosin with ATP with a short delay so that the hydrolysis occurs, and we have myosin in this ADP-PI bound state with its lever up. And then we mix F-actin with it, and we vitrify either after a 10 millisecond or 120 millisecond delay. And that is determined by the distance between the grid as it passes through the spray of the microfluidic mixer and the height of the ethane cup. So here they're very close. That gives us a 10 millisecond resolution. And here that the ethane cup is just moved that little bit further away, and that gives us 120 millisecond timescale. And we chose those because we understand the kinetics of this motor, and we knew, therefore, that at the 10 milliseconds, we're mostly going to trap it in that prime state, whereas at 120 milliseconds, a proportion of that is going to have turned over to a post-power stroke state. And so, therefore, in that second time point, we should have a mix of the two states and any intermediates that may exist in between. So this is about, well, many years on one slide. This is the work of David Klebel, who did all the initial data collection and processing of the cryoEM data. And what David did was to collect 6 data sets, 3 grids on a 10 millisecond time scale, and 3 grids at the 120 millisecond time point. And in that, he could see actomyosin, a free myosin in the background unbound to the actin, and also areas of bare actin. And he was able to perform helical processing, focus 3D classification, and come up with 2 states. So we saw 2 states, one representing a lever up and one representing a lever down, or pre and post power stroke states. And also was able to process the myosin in the background as well to get a myosin structure. And crucially, what we could see when we back calculated the particles and whether they belong to the 10 millisecond or 120 millisecond time point, we found we could see this turnover from this primed lever up state in the 10 millisecond data to the post power stroke lever down state in the 120 millisecond data. And so this resulted in a structure, the first structure of a primed actomyosin state to around 4 angstrom resolution. And this is where Sean came in. So after David did the initial processing and built an initial homology model, Sean then came to refine the model. We're using various tools, Coot, Phoenix, and doing some final processing in Isolde to get this pseudo-atomic model. And crucially, this tells us how myosin works, how it initially associates with actin, which has never been seen before. So we can see that it associates initially with this so-called lower 50 domain through this helix loop helix, and that the upper 50 domain doesn't contact at all. There is some initial association as well with loop 2 and loop 3, which are important if you're a myosin biologist. But the take-home message is that we'd solved the first primed actomyosin state, and we could see that primed myosin binds to actin through its lower 50 domain, with the actin binding cleft open. If we look at what's going on in the nucleotide pocket, we find that we have ADP-PI there, and this back door is closed. And this is a salt bridge that opens to allow for phosphate release. So we know we had a true primed actomyosin state. So you're going to go, okay, great. What does this mean? Why is this important? Well, we wanted to be able to demonstrate the swinging lever hypothesis. So can we now do this with our data? Can we work out the mechanism of force generation? And we also know that actin binding accelerates myosin ATPase activity. So can we explain how? So if you recall in our data, we didn't just have our primed actomyosin state, we also had the post power stroke state and free myosin, which meant that we could actually follow through the cycle in action. So we could go from our unbound prime myosin to see what happens when it binds, to see what happens when it goes through the power stroke to form this post-power stroke state. So the first thing we did, because we knew actin is doing something, and said, is there anything different? And what we noticed was that when myosin binds, the N-terminus of actin suddenly becomes ordered. So this part of actin is reaching out to interact with the myosin. And we could characterize these interactions here occurring with helix W and loop 2 of the myosin motor. The consequence of those bindings seems to be that when myosin binds to actin, the whole upper 50 domain is cocked back relative to the actin axis, which can be seen in this difference map here. And the biggest change is in helix D. And this also results in the upper 50 domain being lifted up away from the actin surface. And when we looked then, what does this helix D change means? It actually induces changes in the nucleotide binding pocket. So now we're getting allosteric effects from binding with actin through to the nucleotide binding pocket, and we can see subtle changes here. And what we think is happening, because upon binding, this helix D is getting shifted back. This is creating more space for the adenosine ring to be pulled back into the pocket, and it's creating strain. Trying to sort of like almost squeeze the phosphate out, but it can't go yet because the back door is not open. And then when we look at the changes that occur upon lever swing, so going from the prime to the post-power stroke state, we can characterize how the lever swings through this 93 degrees, and we can see the changes in the nucleotide pocket when we go from an ADP-PI to an ADP-bound state. Crucially, this allows us to demonstrate the swinging lever hypothesis that is in all your biochemistry textbooks, because we can actually see the turnover from primed to post-power stroke state in our data over time. And if we look at that in a little bit more detail here, you can see that the initial binding interface doesn't change. We've got changes in the upper 50 and the lever position, but that initial binding interface is kept solid. So what we see is when we go from a prime to a post-power stroke state is two vectors of motion. So we have the upper 50 rotating and binding to the actin surface, and then the lever swing. So it's binding of this upper 50 interface at the actin surface that enables a productive power stroke. So that creates the strong binding interface that is needed to resist the force. And it's cleft closure that induces the conformational change in the motor that's needed for the lever swing to occur. So as this cleft closed, the upper 50 binding twists the transducer, straightens the relay helix, and allows the lever to swing. And so it's this upper 50 rotation which moves switch 1 and the P loop away from switch 2 that opens this back door and allows phosphate to exit. And so the question now becomes, what drives that rotation? And if we go back to our N-terminus of actin, we can see there's a change in its conformation between the primed and post-power stroke state. And what's really neat is that in the post-power stroke state, the loop's been shortened. We form this little alpha helix at the C-terminal end. And because the loop connects the two domains, the lower 50 and upper 50 domain, we shorten the loop, we pull on the upper 50. So these initial interactions of this N-terminal extension of actin drive cleft closure, phosphate release, and the productive power stroke. Interestingly, we're only at 4 angstrom resolution. We're making some big claims about what this N-terminal extension was doing. We'd fitted it with Isolde. We thought this density didn't look too bad and this was reasonable, but the reviewers thought we needed some MD simulations. So we did explicit solvent MD simulations in triplicate over 100 nanoseconds, and none of these interactions changed. Yes. There was one very interesting one here, which did look to switch, but when we looked at what was going on, what you were getting here was a change from the association with this oxygen to the alternating one. So these interactions are stable. So with that, we've shown how myosin produces movement and revealed that by time-resolved cryo-EM. Prime myosin binds to actin through its lower 50 domain, with the actin binding cleft open and phosphate release prohibited. It's the N-terminal actin interactions that actually drive this acceleration of myosin ATPase activity by promoting rotation of the upper 50, which closes the actin binding cleft, enables phosphate release, and drives force production. So we've got a little model of it here, what we think might actually be happening. So we've got our myosin coming in here. We have initial loop 2 weak interactions that enable fitting of the helix loop helix in its binding site, largely through hydrophobic interactions. Then we get this little initial cocking back of the upper 50, which allows changes in the end-terminal interaction and closing of the cleft and the productive power stroke to occur. Going to see that from the other side here. So we see that these changes, we get this initial cocking back, and then we get these changes in the actin end-terminus position, which drives this curling up of the loop 2 and upper 50 rotation to allow the productive power stroke. And everybody always asks what's going on in the nucleotide pocket, so we better show that too. So we start with this back door closed. It's still closed with the initial cocking back of the motor, but as the upper 50 rotates, this is opened up, allowing phosphate to come into the phosphate release tunnel, and then the lever to swing. So we've been able to show that actin binding accelerates myosin ATPase activity by promoting cleft closure and phosphate release. And we've demonstrated the swinging lever hypothesis, which was first proposed some 40 years ago. And we have a mechanism for how it generates force. I'm just going to use the next couple of minutes to tell you what's next. So for me, as a myosin biologist, we want to follow myosin in action all the way around the cycle, capturing each of the states. We're particularly interested in trapping the actomyosin ATP state. So this is when you have rigor actomyosin and ATP initially binds. How does it dissociate myosin from actin? And we want to apply this to the study of other myosins in health and disease. As a lab, we're particularly interested in cardiac myosin and its involvement in heart disease. This is being facilitated in collaboration with all the work that's being done in Steve Munch's lab and the sample prep team, where they're trying to make time-resolved cryo-EM easier, faster, and higher resolution with new designs that are modular and easier to use. I'm particularly excited about this sub-millisecond time-resolved cryo-EM that Steve showed you on Wednesday, if you're here, which is using Moses printing to be able to freeze on the sub-millisecond timescale. For us, this is super exciting because this is allowing us to get at this actomyosin ATP state, which because myosin dissociates from actin on the sub-millisecond timescale. So here, the actomyosin ATP state is pretty much all gone by 1 millisecond as the actin increases. But we're actually starting to be able to trap that now. So we're pushing this as much as we can. Also very excited for the speed blot that Steve also introduced on Wednesday. So this is a dot and blot approach, which allows blotting vitrification within about 50 milliseconds. It can do lots of fancy things. I'm particularly interested in how it can be used for time-resolved studies, because the spraying device uses quite a lot of protein. And when you're starting to work on cardiac myosin, which requires mammalian expression, you're not getting so much. Here we can have one of our samples already on the grid, another one on the first set of pads here on a paraffin layer. It mixes very quickly and then blots and freezes. And hopefully you'll see that in that movie here, really quick. It's really cool. And we're using this to look at cardiac actomyosin in action, but in complex with inhibitors. So we do quite a lot of work in my lab, looking at small molecule inhibitors binding to cardiac myosin. How do they bind? How do they affect function? To really understand how they're affecting function, we need to capture them in action. And this is a primed state of cardiac myosin with an inhibitor band that doesn't look like our myosin 5 prime state. So we're really excited about that. So with that, I'd just like to thank everybody that's done the work, the funders, the Asbury Biostructure Laboratory. You can check out this paper on Nature if you here using this QR code if you want, and welcome you to the age of protein cinematography.
Thank you, Charlie, for this amazing molecular insight here. Are there any questions from the audience? Yeah.
Hi.
Hello. How homogeneous were the data at the certain time points? It was homogeneous, or you have different structures each time, and you have to
So interestingly, we're expecting to perhaps trap some intermediate states, and there had been reports of potential intermediate states in this lever swing. But when we did classification of the actomyosin structures, we either saw a prime state or a post-power stroke state. And obviously the lever swing happens on the millisecond time scale. And we think it's rather like the firing of a Roman catapult. So we get that initial little cocking back that allows the rotation, and it's like cocking a gun and then it fires.
One more thing, how do you choose the right time points you want to study?
So I think that's really critical, right? So you need to know a lot about your system and have a very definitive question that you want to answer. So myosin is a really good test case for time-resolved cryo-EM because there is a lot of biochemical data. We have a really good handle on the kinetics. We knew exactly what we were expecting to see. Actually, the ratios we saw of prime to post kind of agreed pretty well with our kinetic analysis of how much we should see at those time points. And it's because we had just such a good handle on the myosin biology.
Okay, thank you. It was a question. Yeah. Whilst you're walking down the stairs, we can actually start with another question, which is related to the first one from Fred Anston from York. Have you tried ATP gamma S instead of ATP to slow down hydrolysis and potentially stabilize intermediate states?
So I think the thing is that we could try that, but it's non-hydrolyzable, right? So we're then trapping it in a non-native sort of state. Yeah. And what we want to actually see is the myosin in action, and capture what's actually going on. So there has been huge amounts of work done in that in the past to stabilize particular states and resolve them by crystallography, which has certainly aided us because without those high resolution structures, we couldn't propose mechanisms in the first place. But we want to actually trap the native molecule in action.
Right, thank you. Okay, now you have a question.
Yeah. A quick question about this actin. Which actin did you use in this studies?
So this is skeletal.
And the human?
Skeletal rabbit actin.
Skeletal rabbit actin.
Yeah.
And so do you think it makes the difference compared to this human actin?
So I don't think it makes a difference compared to human actin, although we have two forms of actin. We use skeletal actin, not the cytoplasmic actin, which actually myosin 5 walks along. But actin is highly conserved. And one of the questions we got, so it's a really good point you've made, because the N-terminus of actin is actually slightly different in the different actin isoforms, but it's still the same charge. So from a DEDE, it goes to EEE or DDD. And so those same interactions we capture can still be recapitulated in these different actins. Otherwise, it's very highly conserved.
Okay, thank you.
Thank you. One more question over there.
Yeah. So through classification, you got two main classes.
Yes.
Now I was wondering, have you done 3D variability analysis so you may capture more dynamic intermediate?
So we did try, and we didn't see anything. That doesn't obviously mean that there isn't anything there. And we are really hoping to do all of this work again. So actually this data was collected some time ago now, and it's collected on our Gaton K2 camera. Now we have 2 Falcon 4 eyes with selectrics. So if we do the experiment again, we expect to see higher resolution. We can collect more particles. We may trap an intermediate, but from all of our analysis, we don't think it's there.
Right. Thank you so much, Charlie, for fantastic talk and discussion.